This morning, it's a particular pleasure, honor, and really privilege to be here today, especially in this very long-standing and venerable Saturday morning physics lecture. This is due to Myron having interacted with me for years now through the Smart Center and thinking of me enough of a biophysicist that he would invite me to a physics lecture. But really, I'm more or less an unabashed chemist. And what I will try to explain to you today is that all things have an underlying chemistry. So maybe building up to Kaplan's talk about all matters physics, I will talk about how everything derives from chemistry. And I will tell you, specifically about some real passion of mine and a hobby of mine, much more about than my actual research, which is on single molecules, as Myron said. And I will try to tell you a story that starts from how chemists can put together various different chemicals here into a round flask and heat them up and have different things happen to them. And they build up into more and more complex molecules, something like this perhaps, that ultimately can give rise to very complex forms like plants, like a rose perhaps, or the tree here, and ultimately explain the tree of life in many ways and explains, for example, where these cute little puppies come from and how we are related to them and why we like them so much perhaps. Okay? Now, I want to ask a number of key questions today. The first question is, what started life, really? Where do we come from? Is a question that, of course, has interested us and humans for millennia. I also want to address the question of what forces drive nature to ever more complex systems, to more and higher complexity as evolution occurs. And I will also give you a brief summary of things, how over the last 6 billion years around the world, we went from unanimated molecules, simple what we call inorganic molecules, to modern biology and human societies. And while this cannot be reenacted and never fully be understood probably to the deepest level, I can connect, and we can connect nowadays, a lot of different points, dots on the map. And I think I will try to tell you what chemists think about how this all happened. I feel obliged to explain a little bit what a chemist is. And I feel that I need to clarify something that seems to be in the popular literature. When you look up mad scientists on the internet with Google, you will find that typically mad scientists are represented as somebody like this. Somebody with a test tube in its hand here. It might explode at any minute. A white lab coat here. Of course, using the face of Einstein, who definitely was a physicist, actually. So more commonly, maybe you find a picture like this, right? A real mad scientist. And again, typically is represented as a chemist here. Or even in the most benign form, you find a guy from CSI, the TV show, right? Still with a very strange, hairdo here, everything on spikes, and using little pipettes and so forth. And you can tell by looking at me that clearly there's a hair density problem here with chemists. So clearly chemists don't all look like this, okay? Any of this for that matter, okay? Now, what can chemists do, right? What can chemists do in terms of explaining the origin of life? And I think what we can start thinking about are two questions that have really been the biggest questions for humanity for a very long time. The one question is, where do we come from, really? What brought us here? And of course, this is connected to the origin of life, to the tree of life, as you see here, where the little chemist here will be next to a poodle, perhaps, and other animals here. And a second question that is related to that, and we also can address with modern techniques, is what makes us who we are, right? There are cells in our bodies, about 100 trillion different cells in our body that make the whole human body here. And how do they all come together? How do they work? How do they find their place within the body to do exactly what they're supposed to do at the time they're supposed to do that? And chemistry can explain all of these. Now, chemistry, of course, you may remember from your chemistry, has to do with molecules. So a water molecule, for example, has 2 hydrogen atoms and an oxygen atom here. But water molecule, other molecules are important too, perhaps a caffeine, right, in order to stay awake during a lecture, perhaps. Or DNA molecules that, of course, contain the blueprint of everything that goes on in our bodies, right? A DNA molecule that has information content. It's really a molecule that stores information. And people think that maybe this is how we will store information instead of hard drives in the future in form of long DNA molecules. Now, connecting this all to the origin of life, we have to, of course, think back of that early time here. And again, you saw that in the movie, an atmosphere with simple molecules in the atmosphere and discharge, I mean, lightning, thunder, and all these things. And how did it really come to this place here? How do we get here? And this is, of course, a representation from a modern biochemistry textbook here, kind of making the connection that there's something that connects us back about 4 billion years ago or so to the original atmosphere on Earth. And in order to understand that, we have to put our Sherlock caps on and think and look at this more deeply with a big magnifying glass and think about what it does. And of course, people have done this for millennia, right? Stonehenge was presumably a way in which people expressed their kind of interest to connect with the gods and to think about how, where we got here, where they got them from. And chemistry can explain a lot of these things, because chemistry can explain what is chemical evolution, can talk about self-organization, and can understand how molecules can self-organize in ever more complex entities. And ultimately also that leads to understanding how biological evolution happens on the organismal level with organisms and animals and all that. Now first, we have to understand what is self-organization. And for this purpose, we have two demonstrations that our demonstrator, Monica, will show us today. The first one is what is called Brownian motion. So what Monica is going to showing us here is a solution of little beads, plastic beads. They are 2 micrometer thin or small in diameter. That's about 1/100th of the width of a human hair. Okay? And the solution, if I showed you here a vial of it, you can see it's a kind of opaque solution, kind of white milky solution here. And we are looking at it here in the microscope. And you see the little beads here. This is really very similar to milk, right? Milk is also a suspension, although in this case it's really an emulsion of little oil droplets in water. So whenever you put your milk into your coffee, you should think about that you're looking at something like this, where little spheres here are the oil, or in this case these plastic beads, are diffusing about and undergoing what is called Brownian motion, which is characterized by this kind of random motion of individual particles around a spot where they are at. And sometimes you see some more directed motion, which is probably a bacterium, quite honestly. I don't know exactly, but I mean, it's something different. But most of these are actually beads, and they kind of hover around in what is called Brownian motion, a motion that characterizes random interactions with the environment, with other molecules that bump into the big beads and push them around, and they go in random circles, basically. It's a random motion, really. And so you might ask, so how can we get any sort of self-organization from this kind of random motion, right? And what we need for that is essentially a second property, which is that these particles interact with one another in some way. And Monica is going to give us a second demonstration that will show this a little bit, using, of course, relevant colors, as she will in a second when she's back, and showing us essentially how a maze and, well, second. Okay, so all blue little caps that have a magnet in them. When they are all blue, they are magnets that repel each other, and you can see that they kind of like in the Brownian motion example, kind of diffuse about and then repel, bump into one another and repel each other and move away from one another. But then when she adds a different polarity of magnet loaded cap here that floats on this water, you can see that actually the yellow or the maize and the blue really come together, right? They really like each other, right? As you see also on your baseball caps. And then she can do kind of magic things with simple rules of interactions, basically. So here, for example, she has some longer magnets on the left and right that attract either the blue or the yellow one, and then you separate them. Again, no magic done other than, I mean, moving some magnets closer or further apart, and then pushing them together, and then they organize. And then now they kind of switch places, right, in this particular case, right? And you can get them to do certain things like this, right? And then when you stir the solution a little bit, so give it a little bit of energy, some waves basically, right? Then you can see eventually the yellow and the blue, the maize and the blue will come again together, because they like each other. They will actually organize into a pattern. And if you stir it a little bit more, eventually you see really like fancy patterns coming out, kind of beads on a string, but you can see that the yellow and the maize and the blue are alternating. But then they can also further organize, and very naturally, through Brownian motion coupled with interactions that they have with one another, can come together in, for example, a rectangle. Right? Right? So they self-organize, as we call this, into a particular shape by their specific interactions that forces them to do that, because the like ones, the like ones repel each other, and the different ones attract each other this way. Okay? So I wanted to thank Monica for her presentation. So give her a hand. And this is the extent of my physics demonstration today, but we will have some chemistry demonstration later too. So, so thanks, Monica. So this brings us back to this question of, okay, if you have now simple molecules in a primordial atmosphere very early on Earth, like we saw in the movie, what will happen? And this question was actually asked by a Nobel laureate of the name of Miller-Urey, who was at the University of Chicago, where actually Myron comes from, I understand. He got the Nobel Prize actually for discovering deuterium, which is important, of course, for the fusion that happens, the nuclear fusion in the sun to produce the sunlight that we benefit from. But after he got the Nobel Prize in 1934, he thought about other things to do, and he got interested in the origin of life. Okay? And one of his students, Stanley Miller here, in his lab did a key experiment in 1953 where he took these same molecules that were thought to be found in this primordial atmosphere here, molecules like nitrogen, which we have around in our air still at 78% today, carbon dioxide, which of course we breathe out and car engines generate and so forth. Also things like hydrogen cyanide, which is actually a toxin nowadays. So this is the ingredient of suicide pills, essentially, that kills people, right? So this is a very toxic molecule, similarly ammonia and other things. And he put them all together into a flask like this. And the water was condensing down here in the round flask, and then he was heating that flask here, and then air or water did evaporate together with all these gases and mixed, and then he exposed it to some energy source, in this case simply a battery-powered spark discharge, and then condensed the water back, and then had that recycling for a few days, for a few weeks, perhaps months. And people have redone these experiments over and over. And when you, again, put on your Sherlock cap and look closely at what's inside, then surprisingly very complex molecules can form from these very simple unanimated inorganic molecules. An example is here something like a molecule that's called formaldehyde, just has an oxygen, a carbon, and two hydrogens here, goes together with this hydrogen cyanide, the toxin that I mentioned, and ammonia, which is also quite toxic nowadays, and they go together and form something that is called aminonitrile here, throws out some water, makes a chemical reaction for a little bit more complicated molecule, and then that condenses with water molecules again to form glycine. And glycine is actually the simplest amino acid that we associate with life on Earth, right? And when you look very carefully at the composition of this discharge experiment, run for a few weeks at a time, you find that glycine is actually quite abundant here. The 4 dots indicate that much of it is found. Another amino acid called alanine, which is the second simplest one, is also found in high quantity. Other acids, organic acids, are found. More complicated ones are a little less abundant typically, but you can see that there's a good abundance of quite a few of these amino acids, aspartic acid, glutamic acid, that make up our proteins in our body right now. Okay? Now, the other thing that's surprising is when the Murchison meteorite a few decades ago fell on Earth and was analyzed for its content of organic matter, a very similar composition was found, where glycine and alanine, again, were very highly abundant, and you can see that the pattern of abundance here looks very similar to that Stanley, Miller, and Miller-Urey experiment, suggesting that on the surface of this meteorite that was flying through space, similar reactions could happen, presumably by the UV light from various different stars. It would pass and so forth. There's enough energy to bring together simple molecules and somewhat more complex molecules that we do know compose modern biomolecules that make up our body. And you can also turn your eye to other reactions. For example, the same hydrogen cyanide here can condense in larger molecules here and eventually form something that we call adenine, which if you know a little bit of DNA, then you know that there are 4 building blocks called A, G, C, and T, and the A stands for adenine. So the very nucleobases that store our information in our DNA, in our cells, is actually made of molecules just like this. Exactly, this molecule is 1/4 of all the information content in our genome, in our bodies that dictate the blueprint of the cell, right? So they can be made from, again, very simple ingredients with a very similar discharge experiment done by Miller-Urey in 1961 at Houston. Now, in principle, one can make a lot of different things. I showed you already that amino acids can be made, and this is again from a biochemistry textbook, just to show you what the chemical structure of glycine would be and what or alanine here would be and other amino acids. And you can tell all of these can be found in these discharge experiments that I just described. Nucleobases like adenine or others, the A and pairs with a T to store genetic information, and G pairs with C, and of the 4 building blocks, they all can be made by similar reactions. And likewise, one can make carbohydrates or sugars, right? Cellulose and sucrose and things like that we would eat, right? Also, very simply from this simple molecule that I mentioned, the formaldehyde with the carbon, the oxygen, and 2 hydrogens here, you condense those together. The sum formula of this is CH2O, right? And you put a few of them together, and then you have a carbohydrate, which has exactly the same sum formula, just more of the same building blocks, right? So you can make all these sugars from it very easily. You can make lipids by a simple reaction from an ethylene molecule here, which is a short hydrocarbon that we would find in the primordial atmosphere together with CO2 makes a fatty acid, which is basically the ingredient for our membranes that Sarah Beach talked about so elegantly last week. Okay? You saw that talk. So very simple chemicals can ultimately give rise to all the ingredients that you will find in a cell. Okay? And the idea to that was developed or was first voiced by Ernst Haeckel in the 19th century. Should give a shout out also to biologists. He was actually a zoologist, and sometimes they can contribute important ideas also. And I know I will have to explain this to my wife later. She's actually a biologist and sits here right in the front row, so there will be a price to pay later, I'm sure. But clearly, the primordial soup might have given rise to these molecules, and then you had all these molecules together, and Haeckel's idea was in this soup they were floating together because they were forming from these simple inorganic molecules to form all these molecules here, and in the soup then they could assemble into more complex things like ultimately cells that we have them in the body now. Now, how can we do this eventually, right? And so far I only showed you how one amino acid would form from such a discharge experiment. But can more complex molecules form, like a protein, for example, that has many of these amino acids knit together, linked together into a long chain? And that's very simple, in fact, because you can take one amino acid and another amino acid, and all you need to do is to do what we say, condense them together, where you remove an OH and an H here together to form an H2O, a water molecule. And so you condense these 2 amino acids to form what we call a dipeptide here, which is basically the simplest form of a protein, if you wish, the smallest form. Of course, normal proteins on our cells are much larger than that, have at least 100 amino acids or longer. But again, by extension, you can just knit them together by these condensation reactions in a very, very simple way. Similarly, condensation reactions also are relevant to link, for example, an adenine here that Miller-Urey made in this experiment in Houston, together with a sugar called ribose, and you link the 2 together, again, expelling water here, and then you make what is called adenosine, so it's adenine with a ribose here, the sugar linked to it. And then you put inorganic phosphate on there again, condensing the two together, throwing out water, and you have what is called a nucleotide, which is the full building block of our DNA, okay, or RNA for that matter, which we'll talk about next. So information materials can be made from very simple condensation reactions of these other molecules that can form from simple inorganic materials. Now, one thing that you might notice is the connecting part here is that in all cases water is being thrown out, right? H2O is being removed in the condensation reaction. And you may say, well, that now I got here, maybe if water can be thrown out, it can also, it's a very high concentration actually in the solution. It's like 55 molar, it's a very high concentration. It can also reverse this reaction. It can, what we call, hydrolytically decay a molecule. For example, it can kind of attack this newly formed bond here and cleave it, and cleave it back into the two amino acids. And wouldn't that be favorable to, because you have such a high concentration of water, and wouldn't everything decay back to a single amino acids? How can you keep a more complex form? And one way to perhaps explain this, and this might resonate particularly with Arborites who have a lot of clay in the soil here, is that perhaps it was clay pockets that have a particular, as a mineral, a particular property of binding amino acid-like molecules, like this one here, which is the glycine that I mentioned here before. And perhaps they could bind them, they bind them pretty tightly actually on their surface. And if you bind two such amino acids together, maybe that could assist in bringing the two amino acids together and to condense them in this fashion here, right? Because they are aligned right next to one another. And this is, in fact, what the ribosome still is doing, aligning these amino acids next to one another and to produce proteins in our bodies nowadays. And so maybe that's what the clay surface was doing here at the beginning of time. And clay, of course, repels water also. And so maybe it kept the water at bay not to decay this dipeptide again, right? So surfaces of minerals might have helped in this regard, is one thought. Now, you may throw something else my way, which is, okay, there's the second law of thermodynamics. Aren't you a chemist? Don't you know about this, right? So the second law of thermodynamics says entropy tends to increase or simply chaos will reign, right? So the idea being that always will the disorder or entropy increase in the universe. And it always has to be either stay level, stay the same, or it has to increase over time. And that's the second law of thermodynamics. That is a law, it has to always be observed, right? So if that's true, then how can we solve that problem of making more complex molecules, right? And so we can see whether I can get this to work. So an example here would be this. So if you put molecules together in one corner, they will distribute throughout the space that they have available. They will not stick together. They will do this, what you see in this little animation. They will go away from one another, out into a bigger space, right? So if you have a primordial soup, maybe you condense something, but then there's so much space to expand into that they will fall apart again and dissipate, essentially, right? Why is that not happening, right? And so there is actually a flaw in that argument because the second law of thermodynamics can be observed as long, and you can also build more complex molecules as long as you have a source for external energy. Okay? Just like in the Urey's Miller experiment, right? They needed a discharge from a spark, right? That's energy, and that energy put into molecules will make them condense into more complex materials. And in general, as long as you can work what we call far from equilibrium and have an energy source to maintain it that way, then you can make more and more complex molecules. Our bodies are, of course, a great examples for that because we have to eat, right, food, and that is ultimately the energy that allows our bodies to maintain ourselves in the shape that we have as long as we live, right? And at the beginning of times, there's some ideas of how this might have happened. Perhaps there was some sort of a geothermal energy migration going on where the molecules that might have been these amino acids or so sunk on the seafloor and then were buried and get closer to hotter areas, close to the upper mantle of our Earth here, and then were synthesized into more complex molecules and then came back up after they had use the energy from down here to basically come back to the seafloor and then ultimately in the ocean to condense further. Another idea is that perhaps the same mineral surfaces, or maybe some other mineral surfaces, allowed some energy to be put into the system. An example for that might be pyrite. I myself am a collector of minerals, so you may have seen this, these beautiful cubic crystals here of this mineral called pyrite. This is a cross-section of that, and it's also called fool's gold, so I try not to fool you today, but it has this golden color, right? It's a very pretty mineral, but maybe it had a special function here also, because the pyrite has a chemical structure of FeS2 and has positive charges on its surface, and might actually also have bound various different organic molecules that sometimes have negative charges on them, and again put them back together in some way, and then maybe injected some energy into them in a form that would allow more complex molecules to build. And I should say this idea goes back to a German here, Günther Wächtershäuser, who was a lawyer, one of the few lawyers who did something useful in their life, I guess. But sorry, hopefully no lawyers are here, so my apologies. But I mean, in a broader sense, you can say that no matter what your background is, you don't have to be a Nobel laureate to perhaps think about the origin of life and come up with some important ideas here, as Wächtershäuser did. So you can redeem yourself always, okay? And, but this idea was further developed by a Nobel laureate, Christian de Duve, who was a biochemist, and again got to think about origin of life questions after he got this Nobel Prize. And he thought about this, that perhaps pyrite or other sulfur-containing molecules here, H2S, hydrogen sulfide, for example, could have condensed with the acids that we talked about that have the COH group here, amino acids have that, and at high temperatures in acidic environments like thermal vents close to underground volcanoes or so, might have formed so-called thioesters here. A thioester being a condensation of a sulfur with the CO group of the acid here. And in fact, thioesters, and this I teach in my biochemistry class, are still a high-energy compound intermediate for many metabolic pathways in our bodies today. So the fact that you have an energy metabolism in the body is in part linked to the existence of these very important thioesters. And so from them you can make ATP, which is another energy source in the body. You can make more complex molecules like RNA. You can make aldehydes and certain sugars, for example, organic acids. So, and that's maintained in our metabolism today. So these pathways exist in our metabolism, in our bodies. And so it's really only one step away from pyrite and organic acids like amino acids binding to the surface to a pyrite crystal here to condense into this thioester that we have as a molecular fossil perhaps in our body still today as an intermediate of our modern metabolism, right? So there are some connections here. Now, what you can build from that are molecules that fundamentally can store information. And the molecule that we work in my lab on particularly are so-called ribonucleic acids, or RNA, and they are related to DNA, very similar, in that they have these building blocks built from an adenine, as I showed, and this ribose sugar that I mentioned before, and then a phosphate group, and you link these together, and you have the 4 different building blocks. A, in this case of RNA, it's a uracil instead of a thymine, and U instead of a T, C and G here. And then you can pair them up in this way, following the Watson-Crick rules, as they are called, after Watson and Crick here. And then you get a double helix like this that looks pretty much like the one on DNA. And I should point out the only difference in our DNA is that this one oxygen here is missing. One atom is missing that's actually removed from the DNA building block, but which starts out as an RNA building block first and then has an enzyme that removes this oxygen in each of these, okay? And so the thought is, and this goes back to Leslie Orgel, that perhaps you can make these information-carrying molecules by condensing these different building blocks together. And once you have them, you have the ability to store information in the sequence that you built. And so if in the primordial soup we had these individual nucleotide building blocks, and then perhaps on the surface of a mineral condense them together, then you have, in the end, a random set of strands of RNA, perhaps. And that's an ingredient where you have a molecule that can store genetic information and where each molecule is a little different from each other because it's perhaps at first a random process by which it assembles in any random sequence, right? And of course, as a caveat here, we are not sure whether this, what we call an RNA world, was first, or maybe there were other polymers or biopolymers that preceded this RNA. But certainly, we can show how RNA might have been one of the very first ingredients of life on Earth. Okay? And this goes back again to Leslie Orgel's ideas, where he said, okay, if you now have these different molecules that have different sequences here, and shown here in red, green, and blue, each are a linear polymer here with random strands of this RNA molecule, and if one of them can do something which is called a template-directed copying, and align following the Watson-Crick rules, so U pairs with A, G pairs with C, and so forth, the building blocks of nucleotides here on the other strands, so to align them, and then is able to fuse them together by this condensation reaction, then it would have made its complement. Okay? And in that way, there's a template-directed copying going on, where the information stored in this molecule would be copied into this molecule as a complement. Okay? And if we had this RNA molecule now had the ability to actually do that itself and act as a catalyst, that is, to accelerate this particular condensation reaction here, right? And then found a way to separate itself from this complement, then you can have this catalyst work on using this complement to make an exact copy of itself, because it would now copy it back into its own sequence here, right? And then perhaps separate this again, right? And then you get more and more strands, and all of a sudden you have a self-replicating property of a molecule, an RNA molecule that was able not just to store information, but to actually copy itself and make many, many copies of itself, and thereby, effectively, by using the energy stored in these nucleotides, overcome the second law of thermodynamics and really make more and more complex molecules using the energy from this, from an external process, essentially, to do that. Now, we know that this might have really been possible because of the work of these two fellows, Tom Cech, bona fide chemist, Sidney Altman, another molecular biologist, actually, that got the Nobel Prize for Chemistry in 1989 for their discovery at the beginning of the 80s of catalytic properties of RNA. So RNA molecules were discovered by these two fellows that do exactly what I just described, to be able to catalyze chemical reactions. Okay? At first, they only discovered ones that are present in our bodies today, in various different cells today. RNA enzymes are quite abundant, it turns out. Our genome has a number of them. The ribosome that builds proteins is a ribozyme, is an RNA enzyme, ribozyme for short, that does knit together these amino acids into a polypeptide, so exactly what I showed you before. And so it's not a big leap to think that perhaps at the beginning of life there were these simple RNA molecules that could make more of themselves and other things to basically build something that is rebuilding itself and making more and more complex forms happen. Now, what you really need for this is three ingredients. You need the ability to self-replicate, right? To take one sequence of RNA that can replicate itself and make copies of itself. Sometimes those copies will not be exact because any copying process will have some errors in it. Right? There's a so-called error frequency that comes with that. And that leads to a sort of mutation here that leads to various different sequences. And you can see that I included this here. Instead of a G, perhaps you get an A here in this particular variant. Or instead of a T here, you get a C in this variant and so forth. So you get various different forms of this RNA enzyme. And some of these RNA enzymes maybe were better than others in catalyzing their self-replication. So through a selection process in the Darwinian sense, so selecting out the fittest, if you wish, of these various different mutants, if we call them, or different species here, would you get one to survive over the others, and that's the one that gets a chance to replicate. at the molecular levels, or we are only talking about molecules, but molecules can presumably replicate in the same way and undergo a cycle of Darwinian chemistry, as I call it here, of replication, mutation, and selection in cycles. Okay? And of course, this is actually from yours truly. This is actually the cover from my PhD thesis a number of years ago. So, but one thing that you, of course, always need, and I just want to remind you of this, is an energy input. Right? You need energy. You need to stay far away from equilibrium because the second law of thermodynamics says that otherwise you would just decay to dust. Right? And so, as long as there's energy to affect this replication and make more of these complex molecules, and energy can come in the form of ATP and other things nowadays, and maybe pyrite in the early days, you can make this cycle go and actually select for a particularly fit replicating molecule. Now, there are other potential obstacles on the way. And one of them is, one thing that you might know about biology is that it always comes in a handedness or chirality. Right? The idea is that something like a protein or a DNA or an RNA molecule oftentimes has kind of a helical character where the right-handed and the left-handed helix are not identical to one another. In fact, they relate like a right hand and a left hand with one another. From your side. Okay, so right hand and left hand with one another. So they are mirror images of one another, but they are not identical. No matter how you put your hands together, you can never make them quite fit over one another, right? And so that's the case for all of biology. You always have these symmetry, this chirality here. You have that in plants that grow kind of in a helical fashion here. You see that in shells, seashells here. Certain helical bacteria look like this. Viruses have kind of a helical character. Even amino acid have a particular chirality to them, everything bigger than a glycine, for example. But even that, so how do you break the symmetry, right? So, I mean, how do you get to one clean form of this? Why is this snail growing its house in this helical form rather than the other? Why is DNA or RNA forming a right-handed helix rather than a left-handed helix, right? But even that can be explained. And even though both forms of these compounds are energetically identical, they have the same energies associated with them, there's a process called symmetry breaking that in the end can lead to an energy well that's like this, where you have multiple different forms perhaps, and this is a kind of artistry rendering of this, where basically there are two forms possible that are isoenergetic, but they are not exactly identical to one another. They have this chirality to them, can either replicate if one wins out by a random event and just makes a little bit more of itself, replicates itself a little bit more effectively than the other, then you get this symmetry breaking where you now start amplifying and self-replicating one form over the other, and eventually that leads to a purification of this one form, because that's the one form that fits to itself and can work more of itself more efficiently because of some random event early in the origin of life that made one just win out by chance, essentially. Okay? And then becomes self-amplifying, and this is how our life nowadays, it has these chiralities fixated in one particular form and not the other. Although we could build, and we have started building, scientists have started building kind of a mirror world of chemicals at least, right? So you could build that. Now, another argument that I sometimes hear when I give this kind of talk is the so-called improbability argument, okay? Which goes something like this. So if you had all these biochemical ingredients, these chemical ingredients that I talked to you about, the amino acids, the nucleotides, and the RNA molecules, and you had also lipids and everything, the sugars all together, and you took them and you got them first from a bacterium, say, that you disrupted entirely, right, and mixed totally so that everything is in random order and there's no relationship in space between things anymore. And now you stirred your pot. You can argue that perhaps the odds that a single bacterium reassembles from just its ingredients would be something like this, an astronomically low probability, 1 in this 10 to the 100 billion or whatever it is, right? So a very low chance. So this is the improbability argument that, I mean, you can't stir it long enough to ever form a bacterium back from its ingredients if you took it completely apart and just put the ingredients back together. But that assumes one thing that's completely wrong. Okay? And the reality check here is to ask whether nature really is working as a blind watchmaker. Right? The idea that you have a blindfolded watchmaker who takes apart the watch and then just randomly fits pieces together, doesn't know anything about the process, and just, tries to hopefully get a working watch back together after a while, will take him a very long time if it just is random. Right? And so that's not how nature works at all. Okay? But this is the assumption under any of these criticisms of this idea that you make more complex forms inevitably. And we actually have evidence for that in our bodies. Okay? And a simple example is that of protein folding. Okay? So in your bodies, there are 100 trillion cells working right now to make more proteins. They make each tens of thousands of proteins each second. Okay? And each of these proteins, obviously, can fold into the specific structure that it needs to fulfill its function inside the cell. This is the so-called protein folding that happens. And it happens very quickly because otherwise we would die, right? So it happens very quickly. The proteins fold into the right form and then fulfill their function within a few minutes, typically, or faster. And why is that, right? How can that happen? And this has a similar there's a similar paradox here, similar to this improbability argument that says, that's named after Leventhal, and that says that even if you make a very short amino acid protein with just 100 amino acids in it, you have a large number of possible conformations. And basically, you connect these amino acids with one another, and they can turn, they can face this way, or that way, or that way, or that way, and you calculate, and then you have another amino acid over here, another amino acid here, so you make a chain. Now you have a lot of different conformations that you have to try out to find the right one. If only one is right, how do you ever find that one if you have this plethora of different, what we call conformations, which is astronomical in a polymer like this, to ever find a proper protein fold that fulfills its function in a cell, right? In principle, just trying out these conformations very quickly will take a long, long time, longer than the life of the universe. And that's because it assumes the following picture, that you basically have a flat golf course, entirely flat. It's a square mile large. Maybe our golf course here applies, but it would be entirely flat and has a single hole just in the middle somewhere, but you don't know where it is, and you're a blindfolded golf player perhaps, and now you're just trying to putt into exactly that hole, and you have no guidance where that hole is. It would take you for a very long time. Okay? Same idea, right? It never can happen. Improbability. But that's not how nature works at all. So nature actually uses an image more like this, where you have a guided landscape. We call this a protein folding landscape. Same thing for RNA also, where basically you have all these different conformations that are represented here on the outside on this rim that all eventually have to make it into what we call the native state, this single state that fulfills its function here. But each of these is following a pathway of its own, a different pathway for every single molecule, but all these pathways go downhill all the time. So it's kind of like you are now again the golf player, but you have the putting hole just at the bottom of a deep well. You basically are putting with a big ball into the Michigan Stadium perhaps, and then you always hit the Michigan Stadium because it goes downhill all the way to the center, right? And so that's how proteins fold in our cells. And this simple thing that we all witness because we are here today, right? All our proteins folded properly, argues why this improbability idea is flawed fundamentally, and why in fact folding into or forming more complex molecular forms ultimately is deterministic and actually inevitable. Now, another way of thinking about this is by thinking about fractals. You might have heard about fractals. They are essentially a complex self-similar geometric structure. Mandelbrot thought about them. So it's a mathematical construct where you basically say that something that is mathematically has a certain so-called feedback loop to generate a color here on a two-dimensional grid. And that color depends on what exactly the calculation gives, and this is a so-called feedback loop calculation. That gives rise to a beautiful picture here, and it turns out that this geometry has a fractal dimension that says that something that when you zoom in on this little part here, for example, it looks pretty much like the big part. And when you zoom in on an even smaller part here, it looks pretty much, again, same like the bigger part. So there's a fractal dimension to it if at various different length scales you see the same general features. And this is actually not a rose, but it's actually a fractal. But you can tell it looks very much like a rose, right? But it's similarly calculated from a complex calculation like this, a mathematical construct, and can form a very complex structure, obviously, from very simple mathematical rules. And you see all these pretty pictures that you can generate this way. Now, how would that work on a chemical basis? And in order to show you a little bit of kind of self-organization, and we are going to have my chemistry demonstration today. You can't imagine how hard it was to cut all the red tape to get Günther Kellner, our chemistry demonstrator, to be able to demonstrate here today. And he has a reaction where he mixes altogether 3 different chemicals in solution, 3 different solutions with one another that he then is going to stir. And there's a stirring bar in here, and that mixes the whole thing up. And the third one goes in there, and it becomes maize, right? And it becomes blue, right? And then it will continue oscillating between these 2 colors, maize and blue, over time. Maize, right? And it will oscillate for a few more minutes or so, going back and forth every few seconds or so. And this is because of simple chemical reactions with a feedback loop similar to ones that you saw in my fractal explanation for the mathematics behind it. And you will be able to continue following this. And I can assure you that my drink today is not this. Okay, so even though it has a similar consistency and color, but it's actually Mountain Dew. Okay, so I'm not drinking that stuff. I'm not that crazy or mad a scientist. Okay. Now, so chemistry can have complex patterns develop from very simple reactions as long as you have some feedback loop and complex underlying reactions. This is called the Briggs-Rauscher reaction in particular. And we as chemists actually have been able to rebuild much of the RNA world one function at a time. These are a bunch of chemists that have done this. So Dave Bartel, for example, came up with a so-called replicase, an RNA replicase molecule. So this is an RNA molecule that can, just as I described, replicate itself. And we can make that by a process similar to what I described from my thesis cover, selection, replication, and mutation. We can evolve that in the test tube. A chemist can have this happen in the test tube, evolve this, and then it does exactly what I described, which probably was starting life on Earth, which is this RNA molecule copying itself into a new molecule. Okay? And of course, you need the energy input, as we discussed. And for that, you need something like a nucleotide synthase, so a molecule that can make the RNA building blocks here in a form that carry energy so that you can condense them together using this replicase. And this was shown by Peter Unruh. And then you can also show, and I mentioned this briefly before, that the ribosome, which was crystallized by Tom Steitz, another Nobel laureate here for chemistry, crystallized by Steitz to show that actually at the core of the ribosome is an RNA enzyme activity that still today knits together these amino acids into polypeptide chains, perhaps like the original clay pockets did in the primordial world. Okay? Now, how can you build more complex things? This is Manfred Eigen, again, another Nobel laureate who, after getting the prize, had nothing better to do to think about the origin of life. And so he came up with some ideas that are called the hypercycle, where the idea is basically that you have a molecule that can replicate itself, perhaps, and make a copy of itself. And if that copy can also help another molecule that then replicates better because of the help that it gets and makes a copy of itself in some form that then goes back and also helps the first one, then you have a little cycle going where the two help each other. right? And that cycle is predicted to be becoming more complex if a third one happens to evolve that now can help either this one or that one replicating and become more complex in its behavior, and then you expand this into a more complex network like this. So hypercycles is the idea that you build bigger and bigger cycles like this, and if you now think that perhaps this doesn't have to be an RNA enzyme that just replicates this, but perhaps makes an ingredient that this guy needs for replication and so forth, then you can build a network of metabolism, just like we have it on our cells today, okay, to maintain us. Okay? Now, another ingredient that you need is to be able to make cellular compartments. You maybe have seen Sarah Veach's talk about how soap bubbles are analogous to the cell membrane that envelopes our cells. And so here, if you just stir these lipid solutions that where the lipids are aligned on the surface of a water-air interface here, and you stir that, and you have some wave action going in this primordial soup perhaps, then you can form what are called micelles, or enclosures that enclose something in the middle here within these lipids, as they are called. And once you can separate self from non-self in this way by enclosing only molecules like these replicating RNA molecules inside a compartment that's enveloped by this lipid here, then you can have evolution and competition between these in a Darwinian sense, where certain ones die out and other ones survive. And that gives rise ultimately to, again, biological evolution now in the Darwinian sense. And so in this way, we believe, and this is now putting those dots together, that RNA was at the beginning of life. There was an RNA world that gave rise to an RNA protein world, where RNA, in the form of an early form of the ribosome perhaps, found a way of making polypeptides of proteins, right, that then folded properly, as was discussed. And then the two together really helped each other and sustained each other, kind of like what I described in the hypercycles. And only much later, DNA evolved when the information content needed to become bigger and bigger. And that we can trace back in our metabolisms nowadays because, as I said, the only difference is the removal of an oxygen from the ribose to make it a deoxyribose for DNA. And you can show that metabolism, this happens by an unusual type of enzyme that probably evolved very late. So the idea is that after the RNA world and then an RNA protein world, we came to our world nowadays of DNA, RNA, and protein. Okay? And so this might run a little counter to what you have seen before, perhaps the so-called central dogma of molecular biology. It says that DNA makes RNA, makes protein. So in this sequence particularly, in the evolution of life, it might have gone differently, that RNA started the whole thing, and then proteins came, and later DNA joined. And in fact, all organisms have, but the central dogma of biology is actually borne out by all organisms. All organisms, from bacteria, archaeobacteria, any animals, everything that we know on Earth has the same chemistry shared. So chemistry is probably what connects us all, because it links to this origin of life and probably goes back to a self-amplification that dates back billions of years. Okay? And so in this way, really, molecules show us where we come from. They are at the root of the evolutionary tree that led to little doggies and humans here. And really tells us that we are all connected in a very, very deep sense, right? We all can trace back our origins billions of years ago to simple molecules that found a way to self-replicate and through a Darwinian chemistry evolve into more and more complex forms. And so we are really latest descendants of this very long tree that has its roots in chemistry. And I mean, Native Americans, of course, had this sense already. If you think about this, they felt a connection between wolves, for example, or any animals for that matter, and themselves. So we are really interconnected with the world around us in a very, very deep sense because of a shared ancestry in form of molecules. And so evolution still happens at all levels today. So we talked about this so far. But of course, our evolution continues. So while we had to think about eat, survive, and reproduce for most part, now we have enough time at our hands to think about where, what this is all about and where we came from and what makes us who we are, right? And evolution of mankind mirrors that of cell phones, right? So evolution is going on at all levels, including in society nowadays, right? And molecules also really fundamentally made us who we are and still make us who we are, because you can zoom in into the human body here and see individual cells light up here with the nucleus and the membrane around here, and things going on in there. If you zoom in, then you see that there are all these molecular entities in there, and when you zoom in further, you see all these molecules diffusing, just in like in our Brownian diffusion example. They diffuse about at any given time and mix with one another and do fancy things. And like, for example, this maize and blue oscillating reaction, which actually resembles our circadian clock, right? The clock in our brain that makes us sleep and wake cycles follow up in 23.5 hours, even if there's no light to tell us that it's day or night. And some may call this divine, right? Now, we also have more evidence that this is happening in our genome nowadays. When the turn of the millennium happened, it was discovered that most of our genome is actually not coding for proteins. Okay? We have 3 billion base pairs, a lot of base pairs, a lot of genetic information, and only 1.2% is coding for proteins. Okay? The rest, as we now know, actually, and this is humans here compared to bacteria in this regard, humans have more than 98% not coding for proteins. Clearly, the more complex the organism is, the more of the genome is not actually serving its purpose on the protein side or on the DNA side, but actually is being transcribed into RNAs, made into RNAs, so-called non-protein coding RNAs, that play a plethora of roles in the body. Again, you won't have to, there was no quiz about this at the end of this. So just to impress you with the idea that RNAs still in our bodies today make everything function the way it's supposed to. They guide everything that goes on in the cell. They make stem cells into differentiated cells, or when you figure out how to do that, you can also reverse this process. They do all of the above. They guide what's going on in our cells, and because they have all these functions. And that probably is in some way a molecular fossil from this RNA world that I described to you today that dates back billions of years to the origin of life, right? And so I could give a whole other talk about what Myron mentioned. We developed this technique, iSherlock, to look at single RNA molecules inside the cell, but this is for another time, okay? Now, take-home messages from today are first, with energy, simple unanimated molecules self-assembled into organic molecules, okay? And these organic molecules assemble into more and more complex molecules like RNA enzymes or ribozymes that can evolve to, for example, copy themselves and really may have started this prebiotic RNA world. And in fact, increased complexity in such a world is inevitable because molecules, based on very simple rules, start assembling with one another. We call this self-assembly into complex patterns. Okay? And they do that because of energetic, going energetically downhill. As long as you give energy to the system, it will become more complex. And our genome, in fact, is then still evidence for the broad utility of RNA and perhaps a remnant in some way of this RNA world that I told you today. Maybe even more important summary is that chemistry rules. Okay? And also, hopefully, I was able to demystify the mad chemist for you today. Right? This is what chemists do, understand fundamental questions of where we came from and what makes us who we are. And really, that brings us all together. Chemists can help understand how we are all linked together, how we are one of all of the same, because our ancestry is all rooted in chemistry, and we still share the chemistry today, which connects us finally back to these cute puppies. Okay, and that's it for me.